Hello and welcome, my dear students and biochemistry learners. Welcome to this video on definition series in biochemistry part 9. In this video, I'll be talking about some very simple and basic definitions related to biological oxidation. So let's begin. Biological oxidation. The transfer of electrons from the reduced coenzymes through the respiratory chains to oxygen, or simply the oxidation reduction reactions or the redox reactions that are occurring in the biological systems. Let's see some basic thermodynamics. Free energy, represented by capital G. The energy actually available to do work, utilizable, is known as free energy. Changes in the free energy are valuable in predicting the feasibility of chemical reactions. The reactions can occur spontaneously if they are accompanied by decrease in free energy, Gibbs free energy. Enthalpy, represented by H. Enthalpy. During a chemical reaction, heat may be released or absorbed. Enthalpy change is a measure of change in heat content of the reactants compared to the products. Entropy, S. Entropy represents a change in the randomness or disorder of reactants and products. Entropy attains a maximum as the reaction approaches equilibrium. The reactions of biological systems involve a temporary decrease in the entropy. Relationship between G, H and S, that is free energy, enthalpy and entropy. The relation between the changes of free energy, enthalpy and entropy is expressed as delta G is equals to delta H minus T delta S, where T is temperature, absolute temperature in Kelvin. Kelvin is 273 plus degree Celsius. Standard free energy. The term standard free energy, represented by delta G0, is often used. It indicates the free energy change when the reactants or the products are at a concentration of 1 mole per liter at pH of 7. Exergenic reactions, obtained from the Greek word exo is outside, ergonic is work. If free energy change is represented by a negative sign, there is a release of free energy. The reaction is said to be exergenic and proceeds spontaneously. Endogenic reactions, andon, within, ergonic work. A positive delta G indicates that energy must be supplied to the reactants. The reaction cannot proceed spontaneously and is endogenic in character. Oxidation and reduction. Oxidation is defined as the loss of electrons and reduction as the gain of electrons. Redox potential. Redox potential of a system is the electron transfer potential, E0. Redox couple. When a substance exists both in the reduced state and in the oxidized state, the pair is called a redox couple. The redox potential, Rp, of this couple is estimated by measuring the electromotive force, EMF, of a sample half cell containing 1 molar solution of both the reductant and the oxidant connected to a standard half cell. Negative redox potential. When a substance has lower affinity for electrons than hydrogen, it has a negative redox potential. Example, NaDH, which is a strong reductant, has a negative redox potential minus 0.32 volts. Positive redox potential. When a substance has higher affinity for electrons than hydrogen, it has a positive redox potential. Example, oxygen, a strong oxidant, has a positive redox potential plus 0.82. High energy compounds. The compounds, when hydrolyzed, will release a large quantity of energy, or the substances which possess sufficient free energy to liberate at least 7 calories per mole at pH 7. Example, delta G0 of phosphoenol pyruvate is around minus 14.8 calories per mole. It's a very high energy compound. Now, enzymes involved in biological oxidation. All the enzymes participating in the biological oxidation belong to the class oxidoreductases. They are further grouped into 4 categories: oxidases, dehydrogenases, hydroperoxidases, and oxygenases. Oxidases. These enzymes catalyze the elimination of hydrogen from the substrates, which is accepted by oxygen to form mostly water. Example, cytochrome oxidase. Aerobic dehydrogenases. These enzymes catalyze the removal of hydrogen from a substrate, and oxygen can act as the acceptor. These enzymes are usually flavoproteins containing either FMN or FAD as prosthetic group, and the product is usually H2O2, hydrogen peroxide. Example, L-amino acid oxidase. Anaerobic dehydrogenases. These enzymes catalyze the removal of hydrogen from a substrate, but oxygen cannot act as the hydrogen acceptor. They therefore require coenzymes like oxidized NAD, NADP, FAD, etc. as acceptor of the hydrogen. When the substrate is oxidized, the coenzyme is reduced. Example, pyruvate dehydrogenase. Hydroperoxidases. Hydrogen peroxide is the substrate for these enzymes. There is a constant production of hydrogen peroxide in the reactions catalyzed by the aerobic dehydrogenases. The harmful effects of hydrogen peroxides are prevented by hydroperoxidases. Example, peroxidase and catalase. Oxygenases. This group of enzymes catalyze the direct incorporation of oxygen into the substrate molecules. They are of 2 types: dioxygenases or true oxygenases. They are responsible for the incorporation of both the atoms of oxygen, O2, into the substrate. Example, homogentisate oxidase. Monooxygenases or mixed function oxidases. They catalyze the incorporation of one atom of oxygen into the substrate while the other atom is reduced to water. So one atom is incorporated into the substrate and the other is reduced to H2O, water. These enzymes are also called as hydroxylases because OH group is incorporated into the substrate. Example, phenylalanine hydroxylase. Electron transport chain or ETC. Electron transport chain or the respiratory chain or the redox chain is a chain of electron carriers, multi-protein complexes, located in the inner membrane of mitochondria. In this, electrons or the reducing equivalents derived from various substrates flow from more electronegative components, that is more negative Rp, to more electropositive components or more positive redox potential in a stepwise manner, ultimately to the oxygen to form water and energy. The different complexes. First, complex 1. Complex 1 or NADH coenzyme Q reductase or NADH dehydrogenase complex collects the pair of electrons from NADH and pass them to coenzyme Q through flavoprotein like FMN and iron sulfur proteins. Energy released from complex 1 is utilized to drive 4 protons out of the matrix of the mitochondria into the intermembrane space. Complex 2. Complex 2 or succinate CoQ reductase or succinate dehydrogenase catalyzes oxidation of succinate to fumarate with simultaneous reduction of FAD to FADH2. Electrons from FADH2 is passed through iron sulfur proteins to coenzyme Q. Enough energy is not released from this complex to pump protons out of the mitochondrial matrix into the intermembrane space. Hence, this may not be the site of ATP production. Coenzyme Q. Coenzyme Q or ubiquinone is a quinone derivative that accepts a pair of electrons from NADH or FADH2 through complex 1 and complex 2, respectively. It gets reduced successively to semiquinone QH and finally to quinol QH2. Electrons from QH2, that is quinol, are then transferred to the complex 3. Complex 3. Complex 3 or coenzyme Q cytochrome C oxidoreductase or cytochrome reductase contains a cluster of iron sulfur proteins, cytochrome B, cytochrome C1 and risky iron sulfur center. It catalyzes the transfer of electrons from QH2 to cytochrome C. Energy released from complex 3 is utilized to pump 4 protons into the intermembrane space. Cytochrome C. Cytochrome C is a peripheral membrane containing protein that is located towards the intermembrane space side on the inner mitochondrial membrane. It contains one heme prosthetic group. It collects electrons from complex 3 and delivers them to complex 4. Complex 4. Complex 4 or cytochrome oxidase catalyzes the transfer of electrons from reduced cytochrome C to molecular oxygen with the concomitant reduction of oxygen to 2 molecules of water. It consists of 4 redox centers, namely heme containing cytochrome A, cytochrome A3, and two copper centers, copper A and copper B. The electron transfers from complex 4 is in the sequence from redox cytochrome C to copper A to cytochrome A, cyto A3, then to copper B, and ultimately to molecular oxygen. Two protons are pumped out into the intermembrane space through this complex. Complex 5. Complex 5 or FOF1 ATPA or FOF1 ATP synthase or simply ATP synthase utilizes proton motive force or PMF for the synthesis of ATP. It is composed of knob and stalk structure like lollipop. The knob F1 is present in the matrix of the mitochondria and has the catalytic subunits for ATP production. The stalk FO has a proton channel and spans the inner mitochondrial membrane. Entries of protons through FO into the mitochondrial matrix help catalyze ATP production by F1. PO ratio. The PO ratio is defined as the number of inorganic phosphate molecule incorporated into ATP for every atom of oxygen consumed. It is a measure of number of ATP molecules formed during the transfer of two electrons through the electron transport chain. When a pair of electrons from NADH reduces an atom of oxygen, 10 protons are pumped out, 3 each from complex 1, complex 3 and 2 from complex 4. 4 protons must flow in to produce 1 ATP through FOF1 particle or FOF1 ATPase. Hence, 10 upon 4 is 2.5 moles of ATPs are formed when NADH reduces an atom of oxygen. So the PO ratio for NADH is 2.5. Simply, 2.5 ATPs are formed when NADH reduces an element of oxygen. And the same is for FADH2 is 1.5 as 6 protons are pumped out. So 6 upon 4 is 1.5. Respiratory control. The regulation of the rate of oxidative phosphorylation by ADP level is called respiratory control. When ATP level is low and ADP level is high, oxidative phosphorylation proceeds at a rapid rate. Oxidative phosphorylation. The process of synthesizing ATP from ADP and inorganic phosphate coupled with the electron transport chain is known as oxidative phosphorylation. Substrate level phosphorylation. Formation of ATP directly coupled to an enzyme formation of ATP directly coupled to an enzyme catalyzed metabolic process without the involvement of the respiratory chain is known as substrate level phosphorylation. That means phosphorylation is occurring at the level of the substrate. Simply, enzyme transfers of phosphate group from a substrate to ADP to form ATP. Example, the conversion of phosphoenol pyruvate to pyruvate is coupled with substrate level phosphorylation catalyzed by enzyme pyruvate kinase. Chemiosmotic hypothesis. Peter Michel in 1961 proposed this theory to explain the mechanism of oxidative phosphorylation. The transport of electrons through ETC is coupled with the translocation of protons across the inner mitochondrial membrane from the matrix to the intermembrane space. The pumping of protons results in the development of an electrochemical or proton gradient. This gradient, also called as proton motive force, drives the synthesis of ATP by ATP synthase complex. Now, inhibitors of ETC. Many site-specific inhibitors of electron transport chain have contributed to the present knowledge of mitochondrial respiration. The inhibitors bind to one of the components of ETC and block the transport of electrons. First, inhibitors from complex 1 to CoQ. That means these inhibitors will prevent the flow of electrons from complex 1 to coenzyme Q. First is rotenone, which is an insecticide and fish poison. Barbiturates, amobarbital, which is a sedative. Chlorpromazine, which is a tranquilizer. And pyridine A, an antibiotic. Alkyl guanides, guanethide, it's a hypotensive drug. From complex 2 to coenzyme Q. Carboxin is a fungicide. Complex 3 to cytochrome C inhibitors. British antileucite or BAL, an antidote of war gas. Antimycin A, an antibiotic. Naphthoquinone, cytotoxic component. Complex 4 inhibitors. Carbon monoxide, cyanide, hydrogen sulfide, and azide. Inhibitors of oxidative phosphorylation or FOF1 ATPase. Oligomycin, an antibiotic. Vancomycin, antifungal. And dicyclohexyl carbodimide or DCCD, which is a peptide coupling reagent. They all block proton flow through FO. Orovatin, an antibiotic, inhibits F1, and atractyloside, which is a plant toxin, inhibits ATP-ADP translocase. Uncouplers. Compounds that can uncouple or delink the electron transport from oxidative phosphorylation. They increase the permeability of inner mitochondrial membrane to protons. They allow electron transport chain to occur but blocks the establishment of proton motive force necessary for ATP production. The net result is that ATP synthesis does not occur and the energy linked with the transport of electrons is dissipated as heat. Example, 2,4-dinitrophenol, 2,4-dinitrocresol, and some physiological uncouplers like thyroxine in high doses and thermogenin, a protein that is present in brown adipose tissue mitochondria. Ionophores. The term ionophore represents the lipophilic substances that promote the transport of ions across biological membranes. They interrupt the development of electrochemical gradient across the inner mitochondrial membrane. Example, valinomycin and nigericin, the antibiotics that act as ionophores for K+ ions, potassium ions. Sorry. Gramicidin, an antibiotic, which is many monovalent cation ionophore. Sorry. Example, sodium and potassium. Excuse me. Oligomycin, an antibiotic, binds with the enzyme ATP synthase and blocks the proton channel. It prevents the re-entry of protons through ATP synthase and inhibits ATP synthesis. Ox-Phos diseases. Disorders involving components of oxidative phosphorylation are referred to as Ox-Phos diseases. Ox-Phos diseases are caused due to the mutation in mitochondrial DNA. Leber's hereditary optic neuropathy (LHON). It is caused by mutation in NADH CoQ reductase, that is R complex 1. Clinical features include blindness, cardiac conduction defects, tremor, and ataxia. Mitochondrial encephalopathy, lactic acidosis, and stroke (MELAS). It is caused due to mutation in mitochondrial DNA gene that encodes for tRNA for amino acid leucine. Clinical features are deafness, myopathy, lactic acidosis, dementia, stroke-like episodes. Myoclonic epilepsy, ragged red fiber disease (MERRF). It is caused due to mutation in the mitochondrial DNA, and the clinical features are abnormal eye movements, myopathy, ragged red muscle fibers in muscles, and progressive dementia. Leigh syndrome is caused due to mutation in mitochondrial DNA and deficiency of enzyme pyruvate dehydrogenase complex. Clinical features of Leigh syndrome is weakness, lack of muscle tone, and movement disorder. Like always, I am thankful to all these resources for making this video possible. Thank you friends, thank you for watching this video. Be happy, healthy, and stay safe. And remember, biochemistry is easy. Thank you so much.